The overall goal of this procedure is to measure changes in membrane properties of olfactory sensory neurons in response to odor stimulants. This is accomplished by first dissecting the olfactory epithelium and transferring it into a perfused chamber. The second step is to locate and choose an OSN of interest under the microscope. The dendritic knob of the chosen OSN is then patch clamped and analyzed while being stimulated with odorants. Ultimately, perforated patch clamping of OSN dendritic knobs characterizes OSN functional properties at the membrane level.
This method can help address key questions in the olfaction fields, such as recording olfactory sensory neurons expressing defined odorant receptors.
And also, patch clamp recordings provide information about the membrane properties of these neurons.
Prepare the stimulating pipettes from 1 mm glass or purchase them pre-pulled. Using a flame, bend 6 pipettes about 1 centimeter from the tip to a 45-degree angle. Then, arrange the 6 bent tip pipettes around a 7th straight pipette, using an eyelet as a guide. And wrap them in heat shrink tubing. Heat the shrink wrap. At the other end of the pipettes, apply more shrink wrap. This makes a pre-pulled 7-barrel stimulating pipette. Now, using a multi-barrel puller, pull the 7-barrel stimulating pipette.